I have a sheep brain, even though it's much smaller than human brain, but we can identify all the structures that we intend to. First, I pay attention to this white, strong layer that is called dura mater. This is one of the three meningeal membranes that cover the brain and protects the brain. And please note that the three meninges of the brain continue with the three meninges of the spinal cord. So whatever we learn about the meninges of brain, we will repeat it when we talk about the spinal cord. Dura mater is made of dense, irregular connective tissue. It's extremely strong. It's full of collagen bundles. And obviously, one of the major things that it does would be protecting the brain. And the middle membrane, the middle layer of meningeal membrane is called arachnoid mater. Arachnoid mater is thin and it's avascular. It's composed of loose connective tissue. So that's one of the major differences, that dura mater is dense, irregular connective tissue, but arachnoid mater mostly is loose connective tissue. It has some collagen and elastic fibers, and more importantly, it's avascular. Between dura mater and arachnoid mater, we have a space that is called subdural space. That space is filled with interstitial fluid. And then when I remove these layers, I get to the deepest layer of meningeal membranes that is called pia mater. Pia mater is attached to the brain. Pia mater is thin, it's transparent, and it actually adheres to the brain. It contains bundles of collagen and some very fine elastic fibers. Pia mater has many blood vessels. Obviously, these blood vessels are used to supply oxygen and nutrients to the brain. Between pia mater and arachnoid mater, we have a space that is called subarachnoid space. That's the space for circulation of cerebrospinal fluid. Recall that cerebrospinal fluid is formed in choroid plexuses that we have in the ventricles of brain. After I dissect the brain, I show you where we look for choroid plexuses, and I explain briefly what they are. But the goal of having cerebrospinal fluid in, for example, subarachnoid space, which is the space below arachnoid mater, or I can say between arachnoid and pia mater, is first of all, cerebrospinal fluid serves as a shock absorbing medium. It creates that optimal chemical environment for neurons to do their activity, which would be generating electrical impulses. And also it exchanges nutrients and waste product between blood and the nervous tissue. Next thing I see, the major part of the brain, I can see the two cerebral hemispheres. And between the two cerebral hemispheres, we have a fissure, we have a deep groove. Right now, that deep groove is completely filled with a very thick layer of dura mater. We name that layer falx cerebri. So we say falx cerebri is part of the dura mater that separates the two cerebral hemispheres. We have another part of the dura mater. This one separates cerebrum from cerebellum. We name this layer of dura mater that I'm pointing at tentorium cerebelli. Between the two cerebellar hemispheres, we have another part of dura mater that separates the two. We call that falx cerebelli. So falx cerebri, tentorium cerebelli, and falx cerebelli. I go to the other side. In sheep, obviously, that's the inferior part. What I see here, I can see the two cerebral hemisphere. The structure that you see here are the olfactory bulb. The olfactory bulb is the place that the olfactory nerves, which we know olfactory nerve forms by the axons of olfactory receptor cells that we have inside the nose. Olfactory nerves pass through the cribriform plate of the ethmoid bone, and eventually when they get here in olfactory bulb, they form synapse, and then olfactory tract is going to transmit the olfactory signals to the cerebral hemisphere. The structure that we see here, these two are optic nerves. Please recall, optic nerves are cranial nerve number 2, but olfactory nerves are cranial nerves number 1. And exactly in this place, some of the axons of each optic nerve crosses and goes to the opposite side. We call this part optic chiasm. When I move posteriorly, I can see this gland, which is pituitary gland. Recall, pituitary gland sits exactly below hypothalamus. So after I dissect the brain through mid-sagittal, we can locate the pituitary gland below hypothalamus. Clearly, that should make sense that secretion from pituitary gland is controlled by the hormones that hypothalamus releases. And then when I move here, I start with brainstem. So recall, part of the brain that is attached to the spinal cord is brainstem, and specifically the part of brainstem that is fused to spinal cord is medulla oblongata. Now I changed the view to the lateral view because I would like you to take a look at, in human we say the anterior of medulla oblongata. We have two bulges. Right now I'm pointing at the right one, but we have also a left one. These two bulges that we have in the anterior of medulla oblongata of a human, we call them pyramids. Pyramids formed by corticospinal fiber tracts, the largest and thickest motor fiber tracts that we have. Those are the fiber tracts that from motor area of cerebrum, cerebral cortex, they transmit signal to the spinal cord to control the voluntary movement of our trunks and limbs. And when corticospinal fiber tracts pass through medulla oblongata, they form the pyramids. And please keep in mind, when we get here, exactly above where medulla oblongata fuses to spinal cord, 90% of each corticospinal fiber tract crosses and moves to the opposite side. That explains why each side of the brain controls the movement of the opposite side of the body. And exactly we name that place that 90% of each corticospinal fiber tract crosses and goes to the opposite side, decussation of pyramid. So that's something interesting to know about medulla oblongata. And please note that medulla oblongata sit very close to foramen magnum of occipital bone. Any sudden movement or applying pressure on medulla oblongata can easily be a life-threatening situation. That's also one of the many reasons that when we want to transport the patient to the hospital, the neck should be stabilized before we start moving the patient to protect the medulla oblongata, simply because in medulla oblongata we do have some very important visceral control centers, neurons that control the visceral activities. For example, in medulla oblongata, we have cardiovascular center. That's the one that controls the force and the number of heartbeats. It also controls the diameter of the blood vessels. So we can say cardiovascular center in medulla oblongata controls also the blood pressure, the blood flow to our tissues. Also in medulla oblongata, we have medullary rhythmicity center, dorsal respiratory center, and ventral respiratory center. They adjust the basic rhythm of breathing. So in short, the neurons that we have in medulla oblongata are the reason for our breathing. That's why any pressure on medulla oblongata could easily stop breathing in that individual. And please don't forget that in medulla oblongata, we also have control centers for some very important physiological processes such as vomiting center, deglutition center, hiccuping, coughing, sneezing center. They're all located in medulla oblongata. So very small part of the brain, but many, many important activities. Above medulla oblongata, we have pons. Recall in pons, we have pontine respiratory group, or some people say pontine respiratory group. The pontine respiratory group with the medullary respiratory group together coordinate our breathing. That's one of the important things to recall about pons. And then above pons, we do have midbrain, which again, I talk about some portions of midbrain after I dissect brain into right and left side. So here is the plan. What I do, I start from the anterior and I go through the most prominent fissure that we have in brain, that is called longitudinal fissure, and I cut brain into right and left side. Looking at the left side of the brain, I dissected brain through mid-sagittal section. First, I follow the spinal cord. Recall, spinal cord is not part of the brain. Brain and spinal cord together form the central nervous system. But I would like you to pay attention to the central canal of the spinal cord. That's one of the places that we look for cerebrospinal fluid. And when I follow this, I can see that eventually I get into a space that is a triangular shaped space between cerebellum. Recall that I said cerebellum looked like a broccoli, and brainstem. that would be one of our ventricles. We call this fourth ventricle. So that's one of the places that we look also for cerebrospinal fluid. And what I do, I get some blood vessels out, but if we analyze this under the microscope, aside from blood capillaries, we find some ependymal cells. Recall, ependymal cells are some of the neuroglial cells. What they do in those blood capillaries that we have here, filtration of blood happens. Filtration means part of the plasma, mostly water and some solute, are pushed out of the capillary. Ependymal cells receive this fluid from blood and then they secrete it into the ventricle. And we name that secretion that originated from blood cerebrospinal fluid. And we name these blood capillaries and the ependymal cells that are covering them together choroid plexuses. So inside the ventricles of brain, we find choroid plexuses. Right now, I'm holding a small piece of one choroid plexus inside the fourth ventricle. Please keep in mind, from fourth ventricle, the cerebrospinal fluid flows into central canal of spinal cord and also flows into subarachnoid spaces that we have in brain and spinal cord. So we find three openings, one in the middle that is called median aperture, and two on both sides, we call them lateral apertures. So through median aperture and lateral apertures, cerebrospinal fluid from fourth ventricle gets into central canal of spinal cord and subarachnoid spaces of brain and spinal cord. From fourth ventricle, we have a long pathway. I'm walking through this pathway. This pathway is called the cerebral aqueduct or aqueduct of midbrain. This pathway is connecting third ventricle that is supposed to be here to fourth ventricle. As I mentioned, fourth ventricle is between cerebellum and brainstem. Third ventricle is between the two sides of thalamus. So right now I am poking the left side of the thalamus. If I place the two sides of thalamus together, I see between the two halves of thalamus, we have a space that is called third ventricle. So that's the job of cerebral aqueduct, to connect third ventricle to fourth ventricle. And then from third ventricle, we have two openings. Each one of them connects the third ventricle to one of the lateral ventricles. So I go inside the cerebrum, deep inside, I find this space. That space is the left lateral ventricle. Recall, we have two lateral ventricles. Each lateral ventricle is located deep inside one cerebral hemisphere. It is connected to the third ventricle by a foramen that we call that interventricular foramen. And then from third ventricle, we have obviously more production of cerebrospinal fluid, because in all these ventricles, we have choroid plexuses. And then fluid goes into cerebral aqueduct, gets inside fourth ventricle, and then here, through median aperture and lateral apertures, gets into central canal of spinal cord and subarachnoid spaces of brain and spinal cord. Now, I go back again to medulla oblongata. One important thing to know about medulla oblongata is that this part of the brain also contains some nuclei that they are associated with several of our cranial nerves. For example, cranial nerves number 12, hypoglossal. Cranial nerves number 11, accessory nerves. Number 10, vagus nerves. Number 9, glossopharyngeal nerves, and number 8, vestibulocochlear nerves. So it's not really surprising that those nerves have connections to the medulla oblongata. When we get to pons, we notice that aside from some nuclei for different activities, which I gave you just one example, like pontine respiratory group, we also have nuclei that are associated to cranial nerves number 8, vestibulocochlear, cranial nerve number 7, facial nerves, number 6, abducens nerves, and number 5, which would be trigeminal nerves. When we get to midbrain, we see that the nuclei that are associated with cranial nerves number 3 and 4 are located here. Number 3 would be oculomotor nerves, and number 4 would be trochlear nerves. So that kind of gives us an idea where we look for the origin of those cranial nerves. And recall that we have also optic nerve that it originates from retina of the eye, and we also have olfactory nerve that it originates inside the nasal cavities in the olfactory epithelium. When I get to midbrain, I notice on the posterior of midbrain, I have two elevations. So I am pointing at these two elevations. We have actually 4 of them. That's 1 and that's 2. These are the left superior and inferior colliculi. That would be the left superior colliculus. This is the left inferior colliculus. The superior colliculi, we have 2 or a pair. They have neurons that they control reflexes that we have to visual stimuli. When we say visual stimuli, for example, by moving your eyes, you track a moving object like a car or somebody passing by, or you're having the scanning movement of your eyes to read a sentence. So that would be one of the things that the superior colliculi do. Recall that they are located on the posterior of the midbrain. The inferior colliculi are pathways for auditory impulses that from inner ear we want to send to the brain. And also in inferior colliculi, we have those neurons that are in charge of reflexes such as startle reflex. When you hear an unexpected loud noise and suddenly you have a sudden movement in your head, trunk, and eyes. So that's the job of the inferior colliculi. And if you pay attention, you see that in sheep, the superior colliculus is much larger than inferior colliculus. Clearly, sheep is the type of animal that respond more to visual stimuli than auditory stimuli. Next, I move to cerebellum, which cerebellum is about 10% of the mass of the brain, but it actually contains about half of the neurons that brain contains. The main function of cerebellum is to evaluate how well movements that our cerebral cortex intended were actually carried out by muscles, and then providing feedback to correct those movements and help maintaining our balance, coordinate our body movement. However, some researchers show that cerebellum also might have a role in some other activities like cognition, reasoning, language processing. So the function of cerebellum is not limited to just evaluating the timing or coordinating body movement. In cerebellum, these elevations of gray matter are called folia, and the white matter that look like branches of a tree would be called arbor vitae. And recall that I mentioned that cerebellum has 2 hemispheres. If I put the 2 sides back together, you can actually see the 2 hemispheres. The central part that connects the 2 sides together is called vermis. And in each hemisphere, we identify 3 lobes: anterior lobe, posterior lobe, and flocculonodular lobe. So that's a short summary about the cerebellum. Now, from cerebellum and brainstem, I move to diencephalon. Recall diencephalon, 80% of it is thalamus. So I'm pointing at the thalamus, and here, exactly this round section is called intermediate mass or interthalamic adhesion. That's the part that connects the 2 sides of thalamus together. Recall, thalamus is grand station of the brain. Many sensory and motor impulses pass through thalamus to reach other sections of the brain. Then from thalamus, when I move anterior and I'm above pituitary gland, I have hypothalamus, which has many, many important jobs. Hypothalamus controls the autonomic nervous system. It secretes different hormones. It controls the secretion from pituitary gland. It's also the thermostat of our body. Basically, it has a significant role in thermoregulation. This is our biological clock. So one of the reasons that human sleeps when it's dark and wakes up when there is light is because of the activity of hypothalamus. Also, hypothalamus contains the neurons that control our eating and drinking. So there is a very long list of functions that this very small section of brain does. So that's about the hypothalamus. Now I go back to thalamus. From thalamus, I go posterior and I can find epithalamus. And the part of epithalamus that in this brain we can see easily is pineal gland. Recall, pineal gland is the one that secretes a hormone named melatonin. During daytime, secretion of melatonin decreases, so we can stay awake. But when it's dark, secretion of melatonin from pineal gland of epithalamus increases, which that leads to sleepiness. And please keep in mind, in the entire brainstem, means midbrain, pons, and medulla oblongata, we have some sensory and motor neurons that we refer to all of them as reticular formation, In reticular formation of brainstem, we have reticular activating system. One of its very important job would be consciousness. In general, damage to reticular activating system leads to coma. And by the way, please note that like any other body part, reticular activating system has many other functions. Here, I just gave you an example of its most important function, which would be consciousness. So now that I learned a little bit about cerebellum, brainstem, and diencephalon, I move to cerebrum, which is the largest part of the brain. In cerebrum, we do have these elevations. We call each elevation a gyrus. So I'm pointing at several gyra. Gyra would be plural. Between the gyra, we have some grooves that we call these grooves sulci. So we have some sulci between the neighboring gyra. We have also some sulci between the lobes that we have in cerebrum. We name the gray matter, the outer gray matter of cerebrum, cerebral cortex. So that's our thinking brain. And deep inside the cerebrum, even though it's not very easy to see in the sheep brain, we do have some other gray matter, which we call that basal nuclei. Recall that basal nuclei have a significant role in sending output to motor areas, for example, to initiate and terminate body movement. If basal nuclei do not receive enough input, for example, from substantia nigra of midbrain, then we end up with a disease such as Parkinson's disease. And as I mentioned, inside the cerebrum, each cerebral hemisphere, we look for one of the lateral ventricles, which, like any other ventricle, contains the choroid plexus. So this was a very short summary of the structures that we can see in brain. I hope you find this helpful.